Welcome to AI Dispatch, your channel for the latest AI videos on YouTube. If you don't want to miss these updates, please like, subscribe, and share. Core Memory podcast features an interview with Nobel Laureate Jennifer Doudna that exposes a counterintuitive principle at the core of modern gene editing. The optimal solution often requires breaking functional systems rather than repairing broken ones. Her work on CRISPR demonstrates that curing sickle cell disease involves deliberately disabling a healthy gene, that viruses represent the ideal medical delivery mechanism, and that editing bacterial DNA inside livestock may prove more effective than modifying the animals themselves. Doudna treats biology as executable code, subject to architectural revision, creating direct conflict with pharmaceutical business models and conventional medical ethics. Standard medical intuition suggests that curing a genetic disease requires identifying the mutation and correcting it at the molecular level. Jennifer Doudna documents a mechanism that operates through opposite logic. The approved CRISPR therapy for sickle cell anemia, called Casgevy, leaves the disease-causing mutation completely untouched. Instead, the treatment targets an unrelated genomic region functioning as a regulatory switch that suppresses fetal hemoglobin production after birth. CRISPR destroys this suppression mechanism, forcing the body to reactivate a dormant hemoglobin system that remains functional throughout life. The cure emerges not from restoration of damaged code, but from strategic demolition of a working biological control system. This principle of therapeutic destruction extends to pathogen utilization. Medical research has historically allocated billions toward viral eradication. Doudna identifies viruses as the solution to gene therapy's primary engineering constraint, delivery precision. The editing tool itself has reached sufficient capability. The bottleneck exists in transporting that tool to target organs without off-target effects. Viruses represent the only naturally occurring entities that have evolved mechanisms for cell-specific targeting at scale. Her proposed solution involves stripping viral genomes of replication capability while preserving their outer protein shells as guided delivery vehicles. The entity classified as a threat becomes the required component for therapeutic deployment. An agricultural application demonstrates a shift in the fundamental unit of biological engineering. Standard approaches to reducing methane emissions from cattle involve selective breeding or dietary modification, treating the cow as the engineering target. Doudna's institute is raising calves where the cow's genome remains unmodified. The editing target is the microbial population residing in the animal's digestive system. Virus-like particles deliver CRISPR components specifically to gut bacteria, reprogramming their metabolic pathways to eliminate methane production. This creates a permanent metabolic change without altering a single nucleotide in the host organism's DNA. The engineering focus transfers from the animal to the microbial ecosystem it contains, redefining the biological boundaries of the system being modified. This permanent modification model creates economic friction with existing pharmaceutical infrastructure. Doudna explicitly states that the industry's revenue structure depends on chronic treatment regimens requiring continuous medication. A single genetic edit that permanently resolves a condition eliminates the patient as a recurring revenue source. CRISPR's technical capability to provide one-time cures places it in structural opposition to the capital incentives driving drug development and deployment. The most effective medical intervention becomes economically undesirable to the institutions responsible for delivering it. Preventive medicine traditionally operates through lifestyle modification or daily medication to manage risk factors. Doudna describes a transition toward preemptive genetic modification in healthy individuals. A person with no symptoms could receive a liver-targeted edit that permanently alters cholesterol metabolism, creating functional immunity to cardiovascular disease or neurodegenerative conditions decades before onset. Medicine shifts from a reactive discipline managing existing pathology to a proactive discipline performing architectural modifications to biological systems before dysfunction occurs. Artificial intelligence is inverting the experimental methodology that has defined biological research. Scientific progress has historically required iterative wet lab experimentation to determine molecular function through trial and error. Doudna observes that AI models now predict protein structure and function with accuracy that renders physical experimentation increasingly redundant. The discovery process reverses. Instead of testing hypotheses to identify functional molecules, researchers will query AI systems to generate optimized molecular designs directly. The experiment becomes a validation step rather than the primary discovery mechanism. Doudna's framework operates from a consistent engineering principle. biological systems exist as modifiable code rather than fixed natural law. Whether destroying genes to activate dormant systems, weaponizing viruses as delivery infrastructure, or editing microbial populations to alter host organism behavior, her approach treats DNA as software subject to optimization rather than sacred text requiring preservation. The emerging technical question: if these optimization strategies prove medically and economically viable, do existing regulatory frameworks and ethical guidelines possess the structural flexibility to govern a world where biology functions as programmable infrastructure? If you're interested in the video featured in this episode, click the link below to watch the original.